Good afternoon. Thank you again for the nice introduction. I'm Giorgia Confalonieri, and together with Paolo Ballerano, today we'll show how X-ray powder diffraction can be applied to modern mineralogy. We'll give a brief introduction to X-ray powder diffraction, what it measures, and how to read a powder pattern. Then we'll move through three short examples: a qualitative analysis focused on phase identification, a quantitative analysis showing how we obtain weight percentages of crystalline and amorphous phases, and a structural example illustrating how diffraction reveals atomic scale information. Let's start to see what a diffractogram is, which is basically the output we obtain from an X-ray powder diffraction experiment. X-ray powder diffraction measures how a monocrystalline or a polycrystalline powder sample diffract X-rays as we scan the angle to theta. So the positions of the diffraction peaks follow the Bragg's law to this sin theta equal n lambda, where lambda is the wavelength. Each set of interatomic planes basically produce a characteristic peak, which is called diffraction peak or Bragg reflection. The resulting pattern represents the sample's fingerprint. So this means that each material has a peculiar diffractogram. So now let's see which are the information that are contained in these diffractograms. So first of all, we have the background that comes from hair scatter, diorder, diffuse scattering due to local disorder, and any amorphous fraction. Treating the background correctly is essential because under or overestimating it can bias phase quantification and mask weak phases. The second information that we can obtain from our diffractogram is the peak positions, because peak positions are related to lattice metrics. This means unit cell parameters and the space group. So basically, with our diffractograms, we can obtain the cell parameters of our sample, and any little shifts in 2 theta of our powder patterns means that we have changes in the cell parameters, maybe due to the thermal expansion, some chemical substitution, or stress. So it's really important to calibrate your instruments to have an accurate calculation of these parameters. So the third things that we have to take into account when we read our diffractogram is the peak intensities, because peak intensities carry the atomic scale information. Through full pattern fitting, which is usually made by Rietveld refinement, we enter atomic position, site occupancies, and thermal parameters. So this is really important for the structural analysis, but we have also to take into account then the intensities in the multi-phase mixtures are related to the phase weight percentages. So this information can help us to quantify the phases that are present in our polycrystalline samples. Last but not least, we have the peak shape, because the peak width and the tails also encode the information of the microstructure. So small coherent domains makes peaks broadly wider across the pattern, while microstain that is related to, for example, lattice distortions, adds broadening as a function of the 2 theta angle. So if we are able to model the instrumental profile, we can actually separate the effect of the domain size and the structural strain, and so get information also about these two parameters. So X-ray powder diffraction enables phase identification, quantitative analysis including amorphous estimation, structure analysis from unit cells to atomic positions, characterization of local disorder, and detection of structural defects via line profile methods. This toolbox spans geology, materials, environmental work, industry, and so on. For all these potential uses of X-ray powder diffraction that we have just introduced, we'll now look at some specific applications. I'll now hand over to my colleague Paolo Baldirano, who will discuss phase identification, the problem of asbestos. So please, Paolo.
Although the term asbestos may appear straightforward, it actually has a well-defined commercial and regulatory meaning. A commonly accepted definition includes 6 fibrous minerals belonging to the amphibole and serpentine groups, which have been or continue to be used for industrial and commercial purposes. Their use is mainly due to their outstanding physico-chemical and technological properties. More specifically, 5 types of amphibole asbestos are recognized: crocidolite, commercially known as blue asbestos and corresponding to the mineral riebeckite; amosite, known as brown asbestos and corresponding to grunerite; and actinolite, anthophyllite, and tremolite, which share their names with the corresponding minerals. In addition, there is one serpentine asbestos, chrysotile, commercially known as white asbestos and corresponding to the mineral chrysotile, one of the 3 naturally occurring serpentine minerals. Chrysotile asbestos account for about 95% of global asbestos use, followed by crocidolite. The remaining types usually occur as contaminants of other asbestos varieties. As previously mentioned, chrysotile belongs to the serpentine mineral group, a subgroup of the phyllosilicates. Minerals in this group consist of alternating layers of SiO4 tetrahedra, the T sheets, and magnesium-centered octahedra, the O sheets. These layers are not perfectly compatible, as the octahedral sheet is slightly larger than the tetrahedral one. This mismatch creates structural strain, which each serpentine polymorph accommodates differently. In the case of chrysotile, the layers curve to relieve the strain, producing elongated, cylindrical, or rolled carpet-like structures that define its fibrous habit. The toxic nature of asbestos was recognized in the 50s. The International Agency for Research on Cancer of the World Health Organization established that asbestos is carcinogenic to humans. In 2005, the European Union introduced a comprehensive ban on the extraction and use of asbestos. To date, 69 countries, representing around 35% of the world's nations, have implemented similar bans, while the remaining countries continue to allow a loosely defined policy of so-called controlled use. Asbestos exposure has been linked to several diseases, including asbestosis, lung carcinoma, malignant mesothelioma, and pleural plaques. The mechanism behind the toxicity of inhaled asbestos fibers are complex and not yet fully understood. One widely accepted explanation involves the presence of transition metals, particularly iron, within the fiber structure. These elements can catalyze the formation of reactive oxygen species, leading to chronic inflammation and oxidative DNA damage. The latency period for asbestos-related diseases is typically long, often ranging from 20 to 40 years. Chrysotile fibers are thought to be less biopersistent and reactive than amphibole asbestos, which may account for differences in their pathogenicity. A few key terms should be clarified. An elongated mineral particle is a mineral fragment with an aspect ratio greater than 3. The aspect ratio is the ratio between the length and width of the fragment. When the width is smaller than 3 micrometers, the particle is considered respirable and classified as a fiber. If the mineral corresponds to one of the 6 regulated asbestos types, it is called a naturally occurring asbestos, NOAA. Otherwise, it is classified as a naturally occurring non-asbestos, NONA. More than 400 mineral species are classified as naturally occurring non-asbestos, some of which show structural and chemical features similar to those of naturally occurring asbestos. Antigorite is one such NONA mineral and a member of the serpentine group. It can easily be mistaken for chrysotile because of their similar composition and appearance. Antigorite displays an alternating wavy structure in which the octahedral sheet is continuous, while the SiO4 tetrahedra are tilted and periodically reverse orientation every half wavelength. The wavelength period can vary depending on crystallographic and environmental conditions. The presentation focuses on a sample of antigorite collected from a vein within the metaophylites outcropping in Calabria, Italy. It is important to note that antigorite, as a naturally occurring non-asbestos mineral, is not currently regulated. Our investigation, which also includes significant geological fieldwork, involves a detailed crystal chemical characterization of antigorite, a test of its biodurability after 1 week in a modified Gamble solution, which is a simulated lung fluid, and an analysis of its chemical reactivity. The main goal of this research is to assess whether the site could represent a potential environmental hazard. The sample was characterized using laboratory X-ray powder diffraction to distinguish between chrysotile and antigorite, to determine its wavelength period, to detect any minor phases, and to identify possible structural modification after the durability test. Electron microprobe analysis, EMPA, was employed to determine the chemical composition, while Mossbauer spectroscopy provided the iron 3+ over iron total ratio. Inductively coupled plasma optical emission spectroscopy was used to measure the cation released by the fibers during the biodurability test, and X-ray photoelectron spectroscopy monitored the surface composition. The diffraction pattern corresponds to pure antigorite, as no reflection attributable to secondary phases were detected. The pattern closely resembled that of chrysotile, except for the presence of weak superstructural reflections, known as satellites, arising from the wavelength modulation. The modulation wavelength was determined from the position of the first to third order weak satellite reflection HS01 located near the intense 001 reflection at approximately 12 degrees to theta. The resulting supercell comprises on average 18 tetrahedra and 17 octahedra, corresponding to the so-called M=18 polysome. The reported crystal chemical formula was obtained by combining the chemical data from EMPA, the iron 3+ over iron total ratio from Mossbauer spectroscopy, and the average polysome type determined through X-ray powder diffraction. The presence of the wavelength causes the omission of some cations compared with an undistorted structure. In this case, as mentioned earlier, the supercell contained 18 tetrahedra and 17 octahedra instead of 18. Additional transmission electron microscopy images and the electron diffraction patterns revealed an extended polysomatic disorder consisting of the coexistence of different polysomes. Antigorite shows higher biodurability than reference Union for International Cancer Control chrysotile. Powder diffraction reveals no structural changes after 1 week in modified Gamble solution, and no precipitates were observed. XPS analysis indicate that iron at the fiber surface is more oxidized than in the bulk. Overall, no significant modification of the surface were detected after the dissolution test. XRPD played a crucial role in identifying and characterizing antigrade. It enabled the determination of the average polysomatic structure and chemical formula and confirmed the absence of minor crystalline phases potentially affecting cation release during dissolution experiments. This mineral shows significant surface reactivity comparable to reference chrysotile. Its radical generation activity is preserved even after leaching in modified Gamble solution. Overall, these results suggest a possible asbestos-like behavior of this fiber.
Now that we've seen how X-ray powder diffraction can be used for phase identification, for example in detecting and distinguishing asbestos minerals, let's move on the next step, the quantitative analysis. In this part, we'll see how X-ray powder diffraction not only tells us which phases are present, but also how much of each one there is. And beyond that, we will look at one of the key challenges in many real materials, the presence of an amorphous fraction, which requires specific methods to quantify accurately. Municipal solid waste is rapidly increasing worldwide. In 2023, it reached about 2.3 billion tons and could approach 3.8 billion by 2050. Landfilling alone is no longer sustainable, both because of limited space and long-term environmental impacts. In this context, waste-to-energy systems play a critical role. They drastically reduce waste volume while generating electricity and heat, thus transforming a problem into a resource. Our case study focuses on the bottom ashes produced at the Turin incineration plant, northwestern Italy. This facility, that you can see here in the picture, converts municipal waste into ash at a ratio of roughly 5:1. So the goal of our work is to characterize these residues and develop a low-cost desanitization method suitable for their specific composition. The TRM Turin plant, active since 2013, treats about 500,000 tons of waste per year through three lines operating at around 1000 degrees. And you can see here a little scheme. So the process produces two types of solids, the fly ashes, which are fine particles from flue gases, and the bottom ashes, which represent the heavier material that accumulates in the furnace. Across Europe, about 21% of incinerated waste becomes bottom ash. So these materials consist mainly of minerals, glassy phases, and metals. In many countries, they are increasingly seen as a secondary raw materials, reused, for example, in road construction or as aggregates in ceramics. Thus, bottom ashes represent truly a challenge and an opportunity because in one way they must be characterized carefully but can be valuable resource if properly managed. Clearly, you can imagine that bottom ashes are not inert. They contain heavy metals like zinc, copper, chromium, and lead, and have a high amorphous content, rising potential leaching risks. European and Italian laws require then a pre-treatment before the reuse to comply with strict leaching limits. Our work focus on the mineralogical characterization of the bottom ashes as a function of particle size, combine X-ray powder diffraction and chemical analysis. This helps link composition, grain size, and leaching behavior, which is essential knowledge to design safe, efficient inertization treatments. Samples were collected directly from the falling stream at the plant, about 20 kilos of materials. The bulk sample was homogenized, dried, and sieved into fraction from less than 0.063 millimeter up to 20 millimeter. And you can see here the curve, ensuring a systematic study of how mineralogy and leaching depend on particle size. We performed chemical analysis using X-ray fluorescence, scanning electron microscopy coupled with EDS and ECP. The main elements are silicon, calcium, aluminium, iron, magnesium, sodium, and potassium. And we can immediately see from this graphic that silicon decreases and calcium increases with decreasing grain size. So we can observe a clear size dependence of the main components in terms of chemical species. By the way, we also detect some heavy metals, which is obviously not a great surprise. And we can see that we have some chromium, titanium, zinc, and copper. However, chemical data alone cannot tell us where these metals are hosted, whether in the stable crystals or in amorphous matrix. So X-ray powder diffraction in this kind of materials is essential. Unfortunately, by the way, with the X-ray powder diffraction, we cannot truly detect the amorphous phase because amorphous has no diffraction peaks. So basically, the amorphous part appears as a broad bump in our diffractograms. You can see here at the bottom a sample without the amorphous, and at the top one which is exactly the same but with an amorphous phase, and you can see clearly the bump. So, in this way, we have to correct our analysis because if we don't take into account amorphous parts, actually the crystalline percentages are often overestimated. So, we need another way to quantify the amorphous phase and correct the percentages of the crystalline phases. The solution to solve this problem is to couple two different methods, the Rietveld and Rear method, which consists in adding a known amount of an internal standard, most often corundum, to the sample before the measurement, the X-ray powder diffraction measurement, obviously. After refinement, we compare the nominal weight percentage of the standard with the value obtained from the fit, and the difference reveals how much of the sample was actually amorphous. Crucial condition, by the way, for the accuracy of this method is that the absorption coefficients of the standard and of the sample phases are comparable. This actually minimizes systematic errors and ensures that the refined ratio are meaningful. Corundum is ideal in our case because it has no peak overlap, no preferred orientation, and its absorption behavior is close to that of common ceramic or geological matrices. With this correction, we can then finally quantify the amorphous content with great accuracy, turning what would otherwise be just a broad bump in the diffractogram into robust numerical information. And actually, this approach is not limited to bottom ashes. It is widely applied in cement, ceramics, glass ceramics, and even pharmaceutical, where amorphous fraction control much of their activity and stability. In this graph here, we can see the mineralogical composition obtained through the Whitwell-Weir method using the X-ray powder diffraction data of the bottom ash. And we can clearly see that it changed a little bit with the grain size. Across all fractions, the amorphous content dominates. So we have an amorphous that exceeds even the 70% by weight. If we take a look On the crystalline phases, by the way, we can notice that the most abundant are quartz, calcite, and feldspar. So it's really similar to the composition of a normal rock, if you want. In the finer fraction, so below about 10 millimeters, we also detect cristobalite, iron oxides, means hematite or magnetite, melilite and alite. The presence of melilite, by the way, it's really important because it confirms us that actually this material was exposed to really high temperatures. By the way, we were not able to find any sulfide phases, and since quartz, calcite, and feldspar cannot host heavy metals in their structure, this means that heavy metals are likely concentrated in the amorphous matrix. It's crucial because this phase, the amorphous one, is very reactive and can therefore release metals into the environment. For this reason, mineralogical analysis must be paired with leaching tests to assess the actual mobility of these elements. Leaching tests were performed according to the European standards with a liquid-solid ratio of 10, and the samples lost about 15–20% of their mass, especially in the finer fraction, confirming, if you want, unfortunately, that the amorphous phase is for sure the most reactive. Here we have the result of the ECP analysis that confirm a clear grain size effect. So you can see that fine fractions release much higher concentration of zinc, copper, nickel, and lead. And copper is the most critical for sure because it's slightly exceeding the legal reuse threshold of 0.05 milligrams per liter, while the other metals remain below limits, but in any case, they show the same decreasing trend with grain size. The pH of the solution was strongly alkaline, which generally limits metal solubility, yet fine fractions still release more metals. This confirms that the amorphous matrix is the main host and source of heavy metals. Overall, finer particles are more reactive and environmentally critical, emphasizing the need for specific treatment of the fine bottom ash fraction. So after the leaching test, we reperform X-ray diffraction analysis on the solid residues because the samples lose about the 15–20% of their mass. The raw diffractogram, so the raw results that you can see here, can be misleading. Phases like quartz or calcite may appear to increase, and obviously these cannot be because no new formation occurs. So to correct this result, we simulated the weight loss and we recalculate phase abundances based on which components actually dissolved. To interpret the X-ray powder diffraction results correctly, we performed a recalculation based on the hypothesis that the phases dissolved during leaching were primarily alide and amorphous fraction. This correction demonstrates that the apparent increase in quartz or calcite was simply an artifact. The recalculation, assuming that the weight loss is due to the dissolution of alide and the amorphous fraction, shows that the crystalline phases remain essentially constant. And this is obviously really good because nothing can be produced during this leaching. This proves, by the way, that the mass reduction is controlled by the preferential dissolution of the amorphous phase, which is also responsible for the mobilization of heavy metals during leaching. Therefore, it is the amorphous matrix that dictates the environmental behavior of the bottom ash. Interesting, in the coarser fraction above 1 millimeter, this phase appears slightly less reactive, likely due to the presence of bottle glass fragments observed also macroscopically, which are more resistant to dissolution compared to the finer amorphous material. To conclude, then the characterization in terms of mineralogical composition of the bottom ashes and the quantification of the amorphous is really important, it's essential. Because the determination of the mineralogical composition, obviously with the chemical analysis, allow us to understand how the heavy metals and the toxic metals are inside our samples and how they will react potentially to the leaching. And this is important for the inertization treatments and the potential reuse strategies. So in these studies, we were able to understand the role of the amorphous phase, since most chemical species exchange actively is controlled by the amorphous indeed, and these paved the way to actually determine new strategies and methods to be able to reuse these materials and not just treat them as waste. Now that we've seen an example of quantitative analysis using X-ray powder diffraction, let's move a step further. In this final part, then, we will see how we can use X-ray powder diffraction for structural determination, which is a little bit more advanced use of this technique. So I will start to introduce zeolites. Maybe you know what is a zeolite, but better to remind, zeolites are aluminum silicates with open 3D framework. You can see here in this little GIF that enables cation exchange, reversible dehydration, adsorption, molecular sealing, and catalysis. They span traditional uses, like, for example, construction, agriculture, petrochemistry, and so on. And some new uses are more advanced uses, like, for example, in gas energy storage, biomedical applications, sensor, and optics. The research output is huge, probably more than 100,000 papers, and mineralogically they are abundant with more than 100 recognized species. So their structural variety makes them both technologically relevant and scientifically captivating, if you want. For crystallographers, for example, they are a continuous source of challenges and insights. One key to zeolite fascination is structural complexity. Frameworks arise from corner-sharing tetrahedra that pack into many architecture. You have to think that we have more than 250 frameworks. So we have this complexity, which is structural if you want, and on the other end, you can add also the chemistry, because obviously you can play with atoms, so you can change silicon with aluminum, for example, and this creates framework charge that can be and must be balanced by extra framework cations. And not only framework cations, but these porosities that you can see here in this little GIF can host also some molecules. So we can host little molecules like CO2, but also something really big like antibiotics. So this coupling between structure, chemistry, porosities actually makes zeolite really important in many different applications. For example, we can think that zeolites and their porosities can be used for the confinement of UV filter, a class of molecules central to the photoprotection. So at Earth's surface, UV comprise 8% of solar irradiance. UVB drives erythema and direct DNA damage. UVA induce oxidative stress, DNA impairment, and skin aging. Both contribute to skin cancers, including melanoma, whose incidence unfortunately is rising. So safer, more efficient UV filters and strategies to stabilize and confine them are therefore critical scientific and social priority. Indeed, UV filters now permeate far more than sunscreens. For example, they are added to plastics, toys, hair care, lipsticks, shampoos, many cosmetics. I'm pretty sure that at home you have some products that contain UV filters. Packaging and printing use them in inks and polymer coatings to prevent fading, often in food packaging where both container and contents benefit of the presence of these UV filters. In short, UV filters are everywhere, so the stability, safety and robust formulations matter across multiple sectors. Unfortunately, by the way, conventional organic filters have limits. Upon UV absorption, inefficient energy dissipation can cause photodegradation and potentially harmful byproducts, reducing protection clearly and raising safety concerns. Complex formulation can foster unwanted interaction or photoallergic reaction. Water exposure accelerates release to rivers and oceans, where persistence and bioaccumulation affect, for example, corals, fish, higher fauna. Links to reef damage have prompted bans or restriction in several regions, for example in Hawaii, in Palau, Thailand, and in many other islands. So improving the stability while minimizing release is urgent, and this motivates the encapsulation strategies of these filters in some other materials. Encapsulating UV filter in porous hosts like zeolites addresses these issues on three fronts. So first, stability, because confinement shields molecules from direct photolysis and reduced degradation pathway. Second, safety, reduced contact and fewer interaction with other ingredients, mitigate irritation and photoallergy while promoting homogeneous dispersion and limiting aggregation. And third, the environment, because physical trapping curbs leaching, lowering bioaccumulation risk, and so in this way we can, in a way, prevent the release of this molecule into the, for example, seawater. For several zeolites with different silicon aluminum ratio and different frameworks, the idea is to find the perfect one to create our hybrid zeo filters. So first of all, we activated with high temperature the hydration, so to remove all of the water from the porosities of the material. Then we load our zeolites with liquid filter at around 100 degrees Celsius for 24 hours, and then we washed everything to remove the molecules that remains, let's say, outside the zeolite. At this point, we were able to use the elemental analysis to quantify the molecules. Again, at this point with the elemental analysis, we are not sure that all of these numbers are related to the molecules inside, because maybe we have some residual outside. By the way, checking this table, you can say perfect phagocytes show us the higher amount of the molecule, so maybe this is the good zeolite. Well, believe me, this is not the good material for us. Take a look on LTL. Despite we have just 0.5 molecule per unit cell, we will see in the next slide that this is the right framework to encapsulate our octinoxate. Indeed, despite the modest elemental loadings, LTA was the most promising zeolite because you can see here UVVs showed superior filtering versus other frameworks. Indeed, other zeolites, like for example the silica mordenite, the silica phagocyte, the ZSM5, unperformed because all of these three last zeolites are acid zeolites and induced even color changes, so it means that we had a protonation of the molecules, and this is clearly something that we don't want because we want to preserve the performance of the molecule or enhance the performance, and this is actually the case of our hybrid material that use the LTL as the framework. So despite we now are sure that our LTL is the perfect zeolite and we were able even to enhance a little bit the performance of the filter, we have some questions. So first of all, is the molecule truly inside? And which is the host-gas interaction between the molecule trapped and the framework? So to answer to all of these questions, we need X-ray powder diffraction, and we also use spectroscopy to couple X-ray powder diffraction with also spectroscopical results. So we performed first of all infrared spectroscopy. We were able to understand through these results that actually the molecules was truly encapsulated into the zeolite because we noticed some shift between the data of the liquid filter and our hybrid materials, and we were also able to see that unlike acid zeolite, which protonate and suppress modes of the filter, LTL preserved the molecular backbone. So now we know that the molecule is inside, but it's really difficult to understand which is the amount of the molecule inside and which is the interaction between the molecule and the zeolite without X-ray. To localize the gas then and to quantify also the molecules, we collect high-resolution X-ray powder diffraction data at synchrotron. The first refinement was done in the normal space group of the zeolite, so an hexagonal space group. We were able to localize the molecule, so we were able to see that there was something inside the porosities of our zeolite, but unfortunately we also noticed some problems in the refinement because we were some misfitting. So basically we tried to reduce the symmetry of our zeolite because it's possible that the presence of the molecule influence also the symmetry of the whole system. So we tried different space group. We did in the orthorhombic, we tried to reduce the symmetry again, and in the end we were able to understand that actually our hybrid material was, it was possible to describe our hybrid material with a monoclinic system. And actually with monoclinic space group, the C2 over M, the fit improved, producing realistic thermal parameters, molecule counts, consistent also with the chemistry that we obtained through elemental analysis, and obviously this proves us that the insertion of the molecules inside subtly lowers the symmetry. We were then able to describe fully the structure of our hybrid material using the C2/M space group. We were able to localize the molecules that now cluster near the BC plane. We notice also a shift of the potassium atoms closer the center of the channel due to the presence of the molecules, and we were also able to quantify the molecules, which is around 0.5 molecules per unit cell. By the way, we can also see that the molecule is not straight along the channel, but actually is a little bit bent, and this is really important because also allow to have longer distance between molecules. But the most important things that we can discover thanks to X-ray powder diffraction is the interaction between the molecules and the framework, because through X-ray powder diffraction now we know that the molecules establish some strong bond with the potassium inside the channels. So it's not just a long interaction with some atoms really far. The molecule is truly stuck inside with a bond with the potassium of our zeolites. To be sure that this bond is real and our model is really close to reality, we performed also Monte Carlo simulation. So we use the model that we designed with X-ray powder diffraction as the input of the Monte Carlo simulation, and actually we can just look at the two images, one from X-ray and the other one with the Monte Carlo simulation. The two models in the end are really similar, and both of the techniques tell us that actually the molecule is bent and it's truly bonded with the potassium of the framework, which is practically the key point of our analysis. Why these results are so important? Well, because we perform leaching tests in seawater, because obviously we want to use this hybrid material, for example, in solar cream. And you can see here that actually the release is really low. And this is because there is this link between the molecule and the potassium in the framework that we discovered thanks to X-ray powder diffraction. Without this, we were not able to understand why these materials have this really nice performance during the leaching test. Thus, the encapsulation in LTL was really successful and simultaneously ensures photochemical stabilization and environmental containment of these molecules that prevents basically the release of possible toxic molecules inside environment. In summary, we were able to prove actually the penetration of the molecules inside the zeolite, which was not so obvious. We were also able to detect some crystallographic changes due to the molecule inside the porosities of our zeolite. We didn't notice any special ordering of the molecules, but we were able to see the number of the molecules inside the porosities, and especially the link between the molecules and the zeolites, that obviously has a really high impact on the application of these hybrid materials. I really hope you enjoyed these three examples. Today we have shown how X-ray powder diffraction can be applied to mineralogy using phase identification, quantitative analysis, and structural determination. These tools let as the code mineral assemblage and track transformation both at ambient and non-ambient condition. In the geological field, X-ray powder diffraction helps to understand processes from formation to changes obviously in some geological processes, but also in industry it helps us to design and tuning materials for technological and environmental application. So in short, X-ray powder diffraction turns complex mineral system into actionable knowledge. On behalf of Paolo Ballirano and myself, we warmly thank Thermo Fisher and Carlotta for inviting us to give this seminar. We have included here the references to the articles behind today's example, so you can explore any topic in greater depth. Thank you all for your participation and attention, and we are now happy to take your questions if you have. Thank you again.